Before I begin this tutorial, what exactly is gene homology analysis and who uses it? Gene homology uses DNA analysis to determine whether organisms share a common ancestor. It's used in the fields of evolutionary and molecular biology, virology, ecology, pharmacy, and anthropology, among many others. First, a DNA alignment algorithm identifies CDS regions that are shared between genomes and translates them into concatenated proteins. After aligning the translated proteins with a multiple sequence alignment algorithm, a phylogenetic tree is built using an algorithm such as RAX, ML, and G. After launching MegAlign Pro, the New Project tab is open by default. Click on Align by Gene Homology. This launches an easy setup wizard. In the reference sequence screen, I'll click Add and add one specific Mycoplasma strain to use as the reference. In the Input Sequences screen, I'll click Add again and add all the strains except the one that I just used as the reference strain. In the Analysis Options screen, I'll leave most settings at their defaults, but for the tree building algorithm, I will specify RAX, ML, and G as the tree building method. This is the method of choice when doing a gene homology analysis. In Assembly Output, I'll type in a project name. If I needed to change the location for saving the project, I could also use the Browse button on this screen. MegAlign Pro is recommending that I run the alignment on my local computer rather than needing to use the cloud. So I'll click on that link to start the alignment. Upon completion of the alignment, the results open automatically as shown here. Let's first look at the phylogenetic tree. Here is the hominous strain that we used as reference, and we can see its relationship to the other strains now. I want this to stand out a little bit, so I'm going to go to the Style panel, and in the Tree section, I will put a box around this with a fill color yellow, and we'll do an outline of red. Before we go look at the sequences view, I'm going to sort those sequences in the same order that we see in the tree. So to do that, I can click on the Sort Sequences button and choose Order Sequences like Tree. We won't see anything happening in this screen, but when we go to the Sequences screen, now these sequences named on the left will be in the same order that we saw them in the tree. To make it easier to analyze the results visually, there are a couple of options I can try from the Multiple Alignment section of the Style panel. One option would be to color only the residues that differ from the comparison sequence. Another option is to choose the Percentage Identity color scheme, which is a great choice for this gene homology workflow. This color scheme uses darker tones to show more conserved regions. At this default zoom level, we can see individual residue level differences between the strains. But if we want to see bigger patterns, we can use the zoom slider to zoom out and get a better view of the features. So here, features represent the concatenated proteins for these different Mycoplasma genomes. Now that we're zoomed out, we can see, for example, that this HU15400175 protein is not nearly as conserved as the proteins on either side of it. If we want to see the gene homology results in the form of a table, we can click on this Homologs button, and I'm just going to make this be a one-panel display so we can see more of it. This version of the table is showing all of the homologs. I can also click here to see homologs that are unique to the reference strain, but in this case, there aren't any. Right now, I'm displaying the function of the gene and its locus tag, and then I have a comparison where I can see the length for the reference sequence and then the other sequences here from genome 1, 2, 3, etc., and percent similarity as well. But I can choose from a wide range of values to display here by moving some over here from the left to the right. I can also see things like the similarity comparison by clicking distance and going to the distance table. And I can arrange these, for instance, ascending by the length. I can also change the values so I can see percent similarity, things like gap length. All of these views, like the sequences view, homologs table, and phylogenetic trees, can be fully customized and exported for printing or for use in a publication.